Hello, welcome. I am the chair of this session, which is on entitled Nanomedicine in and Against Infection and Inflammation. Nice to meet you all virtually. My name is Gert Storm. I'm a professor in Utrecht University, and probably some of you know me already, but let's start quickly. It's not about me, it's about the speaker. And the first speaker in this session is Dr. Nikhil Jain. And he will talk about mechanobiology of macrophage inflammation. And I'm looking forward, Nikhil, to your lecture.
Okay, fantastic. So now, since I'm the first speaker of the session, let me just start by thanking the organizers for the wonderful invitation, for putting up such a nice session, and also for making all the necessary arrangements. And in next 15-20 minutes, what I'm going to show you is how we are using our understanding and our expertise in the field of mechanobiology to better study, diagnose, and hopefully to treat inflammation. Now, as you can see in this live map, we are an aging society. By the end of 2050, there will be more than 2 billion elderly people in the world. And I know I don't need to explain you the kind of socioeconomic burden we will be facing in next 20 to 30 years. And it has become absolutely clear that if we need to fight with this socioeconomic burden, we don't need to fight aging. We don't need to try to reverse aging. But what we need to do is to talk about healthy aging and how we can improve the life quality and the health of the aged people in order to have a healthy aging and also to reduce this socioeconomic burden. So when we talk about aging, it's not about adding another candle onto your birthday cake. It's not about having wrinkled skin. It's not also about having gray hairs. Aging is also associated with multimorbidities, which includes dementia, which includes atherosclerosis, cardiovascular diseases, osteoporosis, and all these different morbidities or all these disease conditions are collectively called as age-associated diseases. And in 2017, more than 92 diseases were identified as age-related. And the way we have so far targeted these diseases is to target them individually. But you can imagine that targeting each and every disease individually, which leads to drug polymorphism, which has more negative effects than the positive or the beneficial health effect. So the question is there any common mechanism among all these diseases, or is there any central molecular player which we can specifically target in order to reduce the ill effect of aging and so as to improve healthy aging? And the answer is maybe yes, we have a central player. It has become absolutely clear that based on the studies conducted by NIH and other several institutes, that as we age in elderly people, there is a dysfunctional immune system, which means that even in the absence of any detectable bacterial infection or any foreign body infection, the immune system in elderly people always remain hyperactivated, which means that the macrophages, even in the absence of any bacterial infection, keep on secreting a huge amount of cytokines. And these cytokines then leads to this excessive secretion of these cytokines then leads to tissue degeneration, and which is right now considered as one of the central reasons for all of these age-associated diseases. And this mechanism or this process is called as inflammaging, which is inflammation during aging. And the way we have, the community has targeted these age-associated cells or aged cells is by either targeting the epigenetics or by targeting the metabolism or by targeting the macromolecular damages. But the central outcome of all these changes is increase in the inflammation. So the question is, instead of targeting all these external all these other events, is it possible if we can directly target inflammation and thereby we could reduce the negative effects of aging? And again, I would say yes, the answer is maybe we can. So the way people have so far targeted inflammation is by targeting the chemical events. So I don't need to explain that when macrophages get activated, though they go into M1 or M2, which is pro-inflammation and anti-inflammation. And I won't get into the detail that once these macrophages get activated, they change their gene expression profile dramatically. But what we found was pretty peculiar, that if you look at these primary macrophages in a in vitro setup, and these are the homeostatic or M0 ground state macrophages, while you can see that they are continuously surveillancing their microenvironment, and while doing so, they are changing their shape, their size, or their morphology dramatically. And we can call them that their geometry or their morphology is more or less random. But they do something interesting when they enter into the process of pro-inflammation or M1. As soon as these cells get the activation signal to enter this pro-inflammatory phase, these cells increase their spreading area to more than 4 to 5-fold, become pancake in their shape, and maintain this architecture or maintain this morphology for long periods of time. Contrary to M1, when these cells enter the process of M2, they, instead of increasing their area or taking a pancake shape, these cells become really polarized, throw these finger-like projections, and again, maintain this morphology for a really long period of time, suggesting that there is not only a transcriptional change during macrophage activation, there is also a morphological change. And to our surprise, if you remove the activation signal, macrophages are known to reverse back to their transcriptional state, their homeostatic transcriptional state, and there is a transcriptional reversibility. But at the same time, we also found that there is a morphological reversibility whereby macrophages move back to their random geometry or their random morphology. And the question is are these changes in the morphology of these macrophages just a byproduct of the activation, or can we use these physical signals in combination with the soluble signals to tune the nuclear events in order to change the inflammatory activation of immune cells, their functional outputs like differentiation and phagocytosis, and so on and so forth. So to answer this question, whether this spreading or the changes in the cell morphology is necessary for macrophage activation, and if you can tune the geometry of the cell and thereby tune the inflammatory activation of the immune cells, we developed these 3 different platforms, all the 3 platforms doing exactly the same thing, confining macrophages in at the single cell level or at the multicellular level. And you can see the first one is a single cell confinement. On the left hand side you have an unconfined cells, on the right side you have a confined cells, and these are the single cell confinement. We also developed sparse cells and dense cells, and you can imagine that in a tissue, the cell-cell contact and cell ECM contact directly control the spreading of the immune cells or any cell type. And then we also developed the quasi 3-dimensional substrates with big pores and small pores. In the big pores, the cells can spread, and the small pores, the cell cannot spread. So for all in all these 3 platforms, we are trying to confine macrophages, and then we ask the question that by confining macrophages, can we tune the inflammatory activation of these cells or not? Surprisingly, if you take these 2-dimensional niches or 3-dimensional niches, and if you culture cells and if you activate them with the very low doses of LPS, which differentiate macrophages towards pro-inflammation, we found that there are 2 different clusters of genes. The first cluster of genes, which we define as primary responsive genes, and they are important for initiating the process of inflammation, were largely similar between unconfined and confined cells. However, if you look at the secondary responsive genes, which include interleukins, chemokines, and iNOS, they were severely downregulated in confined cells as compared to unconfined cells. And as I said, these cluster of genes are important for prolonging the process of inflammation, suggesting that first, spreading of macrophages is required for efficient activation, and second, that if we inhibit the macrophage spreading, or if we confine these macrophages, we can very nicely tune the expression of differential gene clusters. With this observation, the idea was that now, since we see that we can tune the activation by tuning the confinement of macrophages, can we find new molecular mechanisms? And indeed, we were able to. So we found that once these macrophages start to spread, they also start to increase they also start increasing the polymerized state of actin. So as you can see in this plot, in unconfined cells, you have higher levels of F-actin, and in confined cells, you have lower levels of F-actin. And what we now show very precisely that this increase in actin polymerization is absolutely necessary for macrophage phagocytosis properties and also for inflammation. As you can see in this video, you have a macrophage with actin RFP and E. coli in GFP. And as soon as these macrophages try to gobble up these bacteria, they increase the, as soon as these macrophages try to gobble up these bacteria, they increase the polymerized state of actin. And you can see that there is a huge increase in actin structures. And these structures are called as actin pedestals, or they can call it as phagocytic cup. And we are now precisely showing that just by inhibiting this polymerization state of actin, you can actually reduce the phagocytic potential of these cells and also the inflammatory activation of immune cells. And this actin polymerization is not only necessary for eating up the bacteria, it is also important for downstream signaling. Now this transcription factor called MRTFA, it was first found by Treisman at the Creek Institute. And it's an actin-dependent transcription factor. And surprisingly, what we found is that once the macrophages get activated and start the process of actin polymerization, this transcription factor, MRTFA, translocates to the nucleus. So before activation or before an infection, you can see that MRTFA was in the cytoplasm, but as soon as the activation starts or as soon as the actin polymerization starts, MRTFA translocates to the nucleus and get accumulated. And this accumulation results in increasing the expression profile of all the downstream pro-inflammatory genes. And by confinement, we were able to show that in unconfined cells, you have higher levels of nuclear MRTFA as compared to confinement. So again, by keeping the chemical environment absolutely same, just by changing the physical microenvironment of these cells, we were able to change the spatiotemporal organization and the spatiotemporal dynamic of this particular transcription factor, which is necessary for inflammatory activation of the cells. And lastly, we were able to show that indeed upon confinement or indeed in MRTFA knockout cells, or if you depolymerize actin, you reduce the phagocytic potential of the cell and you also reduce their cytokine secretion profile, and thereby you can nicely tune the activation of immune cells. And our hypothesis is that in healthy tissues where you don't need immune cells to be activated, cell-cell contact and cell ECM contact keep the spreading of these macrophages and thereby control their activation potential, whereby whereas if you have any kind of tissue damage or if you have any kind of wound, the macrophages get the chance to spread and increase their inflammation and thereby help in tissue repair or wound healing. And these observations not only have this clinical benefit, but also biomedical importance, because we know that as soon as we put any kind of bioimplant in human body, macrophages access these bioimplants, consider them as foreign body, and start the process of inflammation, and thereby reduce the efficiency and the efficacy of these bioimplants. And the idea is, can we design bioimplants having different pore sizes, where we know that macrophages will still access these bioimplants, but will not be able to initiate sufficiently the process of inflammation, and thereby we can increase the lifetime of these bioimplants. We don't know that, maybe it's true. Now with this, I'll change gears a little bit and I'll talk about epigenetics. We know that these transcription factors, which I have just spoken about, moves to the nucleus, and once they're in the nucleus, they need to bind to their promoter regions. And to do that, there needs to be a huge amount of chromatin remodeling, which is necessary to open up the DNA or to open up the chromatin. And these DNA epigenetic modifications are becoming more and more important in the field of chronological aging and the biological aging. And we know that when you have more methylation of these genes in the promoter region, you have lowering of gene expression. If you have higher demethylation, you have higher expression of gene expression. But the field is currently struggling with the inflammation field is currently struggling with the techniques that are available to quantify or to look at the DNA epigenetic modifications, because the techniques which are available are not fast. Techniques like bisulfite sequencing are very costly. They are not very sensitive. And most importantly, they also need huge amount of starting DNA material. And to answer this to overcome this problem, we started collaborating with Yuval Ebenstein, who is at the Tel Aviv University. He's a chemist who is interested in developing single molecule techniques to look at the DNA epigenetic modification. I won't get into the detail of the technique, but the technique involves isolation or isolating long stranded of DNA from the tissue or the cells of your interest from any organism, mouse or human, by using specific DNA methyltransferases and modified nucleoside analogs. We can specifically fluorescently label each and every MC or 5-HMC or these DNA epigenetic sites. And once you stretch the labeled DNA in the microfluidics channel, you can see the DNA looks like these long pieces of thread. And these threads have these beads or these fluorescently labeled tag for each and every epigenetic modification. And this is how the DNA look in real time. So you have green color showing the DNA and the red color showing each and every epigenetic modification. You just need to grab thousands and thousands of images, feed it in a software which is custom built. The software will detect each and every individual DNA molecule, each and every DNA epigenetic modification mark. It will quantify for you the global levels in the changes and the global levels of modifications, the epigenetic modification at the level of methylation, hydroxymethylation, and non-methylated cytosines. And with this technique, we were able to show that once macrophages get activated, they increase the levels of hydroxymethylation, decrease the levels of methylation, and also increase the levels of unmethylated cytosines or the unmethylated promoter regions. And I won't get into the detail, but we have now very carefully characterized what all the critical DNA methyltransferases and the enzymes which are responsible for macrophage activation and the changes in the DNA epigenetic modifications, and how we can specifically target them to reduce it. And the next 2 minutes, I'll talk about this one last observation, which we are really excited about. So I have shown you that you can tune inflammation by targeting actin filaments. You can tune inflammation by targeting the chromatin epigenetic modification. But these two compartments or these two parameters are not independent. They are physically connected. So if you look at actin filaments, they are physically connected with the chromatin via these nuclear envelope proteins, which have a cytoplasmic domain on one side to bind to the actin, and the nucleoplasmic domain which binds to the chromatin. And one of these central nuclear envelope protein or this linking protein are the lamins, which are known to help in maintaining the mechanical stability of the nucleus, help in maintaining the chromosome organization, anchoring transcription factor, and has been extensively studied in progeria and human aging, but not in the context of inflammation. And we found that recently that once macrophages get activated, as you can see in these immunofluorescence images, there is a significant reduction in the levels of Lamin-AC. So in an homeostatic macrophage or M0 macrophage, you have higher levels of Lamin, whereas in LPS treatment, you have lower levels of Lamin. And this observation was not only specific to bone marrow-derived macrophages, but to any tissue-resident macrophage. If you take alveolar, microglia, or peritoneal, and once you activate them, there is an increase in pro-inflammatory genes, but there is a significant reduction in the levels of Lamin. The observation is also independent of the strain of the mice, whether it's C57 or BALB-C. It's also independent of whether you are using human macrophages or mouse macrophages, cell line, or whether you are using primary cells, showing that this observation is pretty generalizable. And the question is, now can we target these Lamin downregulation in order to change the inflammatory activation of immune cells? I won't get into the detail of all the drugs because of the time constraints, but now we have shown, and the work has been done by Johanna Mell, a fantastic graduate student, that if we inhibit the phosphorylation of Lamin, or if we inhibit the degradation of Lamin, or if we inhibit the downregulation of Lamin by targeting these different events, we can basically reduce the inflammatory activation of immune cells. We reduce all the metabolites which are necessary for inflammation, and overactivation of these macrophages, which leads to loss of function, can be reduced by targeting most of these events like phosphorylation of Lamin or the degradation of Lamin. And at the same time, since Lamin provide the mechanical stability to the nucleus, we are also working in the direction to see whether we can design or we can use nuclear lamina as a biophysical diagnostic marker for macrophage activation. So to wrap up my talk today, I've shown you that it's a well-known fact that as we age, there is an increase in inflammation. And the mechanobiology community has now started to look at in the cytoplasmic side what are the critical mechanosensitive elements like actin, in the nucleoplasmic side like lamin and chromosome organization, and how these two compartments and their mechanosensitive components changes as we age. But what we still need to establish is a functional correlation and a regulatory correlation between inflammation and aging, and also the mechanobiology. And our understanding is, and our aim is to see if we can target these very specific mechanosensitive components in the cells in order to reduce the inflammation and improve what I call as healthy aging. And lastly, the aim is, can we establish mechanobiology as the 8th pillar of aging? And with this, I want to add that the work was done in Viola Vogel's lab. I really want to thank our collaborators and our funding agencies. And with this, would be happy to take questions. Thank you very much.
Thank you for a very nice starting lecture, setting the tone probably for the whole session, because we are probably discussing mechanobiology in relation to immunology and immune effects more deeply in this session. I have to look whether I have some questions, and I have to see whether I can find the questions. Wait a second. There are no questions yet. What I am, maybe I start off, and most of the questions we will do after the session. We have 1 minute. Maybe this is the question. If I listen to you, I think this whole concept is highly intriguing, and it's step by step. It's going in a very good direction. But if I read all this, this is all towards pro-inflammatory effects. So at the actin levels, mechanic effects on the nucleus, what you can maybe monitor and see. And suddenly at the end, I see the word anti-inflammatory effects, which are maybe even more, but maybe you first have to detect the pro-inflammatory effects. Then you can maybe think of therapeutic ways to change to anti-inflammatory effects, which would be highly beneficial to the patients. Can you comment on this in general?
Gert, Beat here. You have questions. There are 3 questions.
Okay.
You have to refresh the red button, and you have to refresh.
Okay, then I maybe he answers my first question, and I go to see the questions.
Yes, you know how. Moderator 1, 2, 3, 4.
Okay.
In the orange button.
Okay.
Okay. So can I answer?
Yeah, you can answer, please.
Yeah, okay. So what you're asking is that whether we are more looking towards pro-inflammation or anti-inflammation.
Yes.
So what we have done so far is we have now find out what are the new molecular mechanisms by which we can tune the pro-inflammation. And when I use the word tuning the pro-inflammation, the idea is how we can reduce it. And whether it's by confinement, we can tune or we can reduce the inflammation. And again, in the last part of my talk, I mentioned about the nuclear lamina. And there also the aim is how can we target these molecular mechanisms like nuclear lamina degradation in order to reduce the pro-inflammation, which is to a certain extent, you can call it anti-inflammation also, because the idea is to reduce the inflammation by targeting these molecular mechanisms. And that's why I have always spoken about hyperactivation of immune cells and how we can reduce this hyperactivation.
Okay, I think that's a very clear answer. I must say I have problems to find the questions, but the agreement is that we have 20-minute talks, and we have passed our 20 minutes, and we proceed with the second talk. In the meantime, I will try to find the questions, and we will do that at the end of the session. We will go through the questions.
Fantastic.
Thank you so much. Very clear. And let's go to Dr. Jordan. And he is from Geneva, at the Institute of Pharmaceutical Sciences of Western Switzerland, University of Geneva. And he will talk about nanocrystals and osteoarthritis treatment. Please take the floor, the virtual floor.
Yes, good morning, ladies and gentlemen. I'm pleased to present you some work we performed in the School of Pharmacy in Geneva. Sorry, I just have to. Yes, on a different field. Actually, we'll take you to a world combining nano world and macro world, showing you nanocrystals, polymer particles as sustained drug delivery vectors for osteoarthritis treatment. So some key points for osteoarthritis. It's a highly prevalent disease that affects the majority of the elderly population. The risk factors beside age and genetics are overweight, joint trauma, and overuse. There is no yet efficient cure available, and the current treatments essentially manage the symptoms. They most often aim at reduction of inflammation and pain. I'd like to show you this video of a normal knee joint with wide cartilage surfaces and stabilizing ligaments. On the right side, you see the joint undergoing cartilage degradation, typical of an osteoarthritic disease. So an attractive option to reduce systemic effects is to inject locally the active drug through a procedure called intra-articular injection. So we're going to focus in the talk on intra-articular injection of a drug delivery system. We need to comply with some requirements. First, osteoarthritis is a very slowly progressing disease, taking years between the onset of the disease towards a full cartilage degradation. Intra-articular injection may be repeated every 3 months. However, more frequent injection is not desired since they would reduce patient adherence to the treatment. They could increase the risk of infection, the treatment cost, for instance. So given this, we identified two main requirements for intra-articular drug delivery. First, we need an extended availability of the drug in the knee. And as a consequence, for a long-lasting therapeutic effect, we will need also relatively high drug doses to sustain the effect. Two different approaches may be mentioned. First, the suspension of drug crystal. For instance, if you have a pain in the joint, your doctor may prescribe an intra-articular corticosteroid shot, which is actually a suspension of drug particles. Such particles could be milled down to nano size, resulting in so-called nanocrystals, which will stabilize with surfactants. Here, the high crystalline molecular packing allows to deliver high drug dose. Also, this solution occurs in a relatively fast way within hours to days. Another approach is to use biodegradable polymer particles, such as a polylactic glycolic acid, typically, and the slow polymer degradation allows to release over several months. Still, the usual emulsion evaporation process used to produce such particles lead to a relatively low drug content. So now how to combine a high drug dose with a slow release? We hypothesize that the combination of these two approaches could accumulate the advantage of the two techniques. That is, embedding drug nanocrystals into biodegradable polymer particles using local administration to reduce side effects. And we then should choose a particle size which is adapted to the long-term joint residence. So for this later point, I would make a quick digression, showing you a primary study where we investigated the fate of macroparticles of different size, 300 nanometer, 3 micron, and 10 micron, which were injected intra-articularly in joints of nice, healthy joints, or arthritic inflamed joints, which are more permeable to particles. We made the biodistribution at 6 weeks. The 300 nanometer particles were washed away relatively fast, whereas the 3 micron stayed in the healthy joint, but not in the inflamed joint, where transported through lymphatic circulation vascular vessels. Only the 10 micron larger particles stayed in the pathological joint. So we selected large particle of at least 10 micron as a target size. So now that we have designed our drug delivery system, we need to check our hypothesis of sustained high dose delivery. through specific steps. First, to produce the stable suspension of nanocrystals, to incorporate these nanocrystals into larger macroparticles, then to characterize these particles on their drug release. I will show you the example of an anti-inflammatory celecoxib. And finally, to establish a preclinical proof of concept in an osteoarthritic model, I will show you the example of a chondroprotective drug, cartogeny. So first, producing stable suspension. We start from the crude amorphous drug, which is first purified and crystallized using the solvent non-solvent precipitation method, which leads to a pure, highly crystalline compound, which is, however, made of very large crystals. So we need to reduce the size in a top-down approach by wet milling. We reduce the size down to 300 nanometer nanocrystals. And in order to prevent aggregation and to inhibit crystal growth, we need to stabilize with surfactant, and we choose well-characterized surfactant, vitamin E TPGS, that has a long track record of safety. So we have our suspension of nanoparticles. How to incorporate them in macroparticle? We use the spray drying techniques. So we started from a suspension of nanocrystal in a polymer solution that we injected into the spray dryer. Spray drying consists in first atomizing the particles, and the liquid polymer droplets are dried into and precipitated into macroparticles, which are collected and then further lyophilized. At the end, we got our nanocrystal polymer particles. So I will not detail the optimization of the process parameters, such as polymer concentration, feed rate, atomization pressure, and temperature, which we undertook in order to decrease polymer loss and obtain a reproducible particle. I would just say that one advantage of this spray drying technique is that it's a continuous process. And since it's not in solvent, it's in air, you do not lose drug. You have encapsulation efficiency rather high, typically higher than 90%. So we have our particles, and actually I'd like to show you now the characterization of these particles using a specific drug. It is celecoxib. It's a selective cyclooxygenase 2 inhibitor, an anti-inflammatory drug. It's commonly used, prescribed in the symptomatic oral treatment of osteoarthritis. It's an alternative to corticosteroids. So we produce two types of particles. First, the drug solubilizing particles, which are on this top row. On the lower row shows the nanomil drug in particles. All particles are the median diameter in the range of 30 micron. We measured the drug content, which was by UPLC, which was 25% for this first column or 50% for this second column. The cross-section of the particles shows a smooth polymer matrix for the drug solubilized, as well as the absence of crystallinity as measured by differential scaling calorimetry and confirmed by the absence of peak on the X-ray diffraction pattern. In contrast with the nanomil drug in particle, we show an inhomogeneous cross-section with some crystals or crystal aggregate with crystallinity of 24% for the 25% drug loading and 70% for the 50% drug loading. And crystallinity was confirmed by the appearance of peak of celecoxib in the XRD pattern. So for the drug release, we assess this in a phosphate buffer saline with 0.5% SDS in order to have sink condition at 37 degrees. We saw no burst effect for all the condition, all the drug loading on the two types of particles. But clearly, over the 3 months of the study, we had a much slower release for the nanoparticle in polymer, nanocrystal in polymer particles compared to the drug solubilized particles. So extended release over several months. In terms of preclinical proof, we selected another principle, active principle, that is cartogeny, which may act actually on the progression of the disease. That's what we call disease modifier. Cartogeny is able to induce chondrogenesis of stem cells and of cartilage cells also by upregulating collagen 2, aggregants, and tissue inhibitor of matrix metalloproteinase. So with this cartogeny, we made, again, nanocrystal in the same way. We use a fluorescent polymer to produce macroparticles, 31% drug loading, high drug loading, and this time a bit smaller, 14 micron. and with also an extended release again over 3 months. These particles were then assessed in vivo. The efficacy was assessed in an intra-articular model of destabilization of the median meniscus, the so-called DMM model in mice. So this model, it involves the meniscus, which is commonly affected in human osteoarthritis, and it mimics the progression of the disease as seen in humans. The timeline was to destabilize the meniscus at day 0, and then to inject the first shot of the NPPs at week 1, a second shot at month 1, and the sacrifice at month 2. We use 6 weeks per group. The result here, the animation shows the intravital fluorescence of the macroparticles superimposed with X-ray microtomography at 2 months. This suggests the permanence of the microparticle in the joint. And if we look at the fluorescence, the mean, the average signal is constant over the 2 months, which suggests that most of the macroparticles were still present in the joint at 2 months. And if we now make histological cross-section to find the particle, we can observe those microspheres in HA classical histological sections. They are mainly observed as trapped in the lateral synovium. And we know these are macroparticles because they are fluorescent tagged, and we can see on fluorescence the red dots corresponding to the particles. So a specific localization in the synovium of these large particles. Now, if we look at the outcome in terms of bone, first, macrotomography at 2 months allowed to measure the ratio of the epiphysis. So this is the epiphysis, and the medial thickness here compared to the lateral thickness is a marker of the progression of the disease. It is reduced actually compared to the non-operated healthy joint. This medial versus lateral ratio is reduced for a pathological osteoarthritic knee. If we use blank particles, it doesn't help to improve. And if we use solution at the same concentration nanoparticles injected at months 1 and 2, it also doesn't help to improve the picture. However, with the particle, we see a trend towards an improvement, a protection of the epiphysis at 2 months. If I have time, I may also go for the histology.
Yeah, 2 minutes more.
Okay, so that's the last slide. So we performed the histopathological analysis, tissue section were stained with safraning O and fast green. This shows the medial side of the knee of the mice at 2 months. So you can see the femur above, as well as the tibia below. And in purple, you can see the cartilage, which is stained here. The arrows indicate the cartilage erosion that you can see in the OA model. Now, for the condition of the treatment, the blank particle did not impede the observation of cartilage degradation. The cartogenic solution at the same concentration did not allow either to improve this picture. Only the cartogenic nanocrystal polymer particles could preserve the cartilage smoothness at 2 months. We actually, so these are only histology, we actually performed also scoring, the ORRSI scoring, to prove that we had a significant improvement with the cartogenic particles for the medial tibia, medial femur, medial cartilage erosion, medial cartilage scores when compared to the osteoarthritic condition. So we conclude that this high drug dose provided by these cartogenic nanoparticles enclosed by macroparticles enabled cartilage protection and regeneration, maintaining the effective drug concentration despite of joint clearance. And to conclude, we combine wet meal nanocrystal with polymer macroparticles to allow extended release of high drug payloads. We may apply that for a whole range of drugs. We also did that for dexamethasone or a MAP kinase inhibitor, PH38. However, for each new drug, we have to make a new specific optimization and development. I showed the preclinical proof of concept of a potent osteoarthritis disease modifier. And besides working with new molecules in the future, we'd like to rationalize our approach, which could be supported by in silico modeling of these particles. I'd like finally to thank the group of the Institute of Pharmaceutical Science of Western Switzerland, the School of Pharmacy at the University of Geneva, and specifically Professor Aleman and our doctors or PhD, Pierre Maudin, Carlota Salgado, and Julie Pradal, who performed the work that I've shown, as well as our clinical collaborators, Professor Gabet and Anoush from the Geneva University Hospital, and you for your kind attention. Thank you.
Thank you for, let's see, am I unmuted? No, I'm muted for your talk, interesting talk, and describing the current status. I do the same with this talk we did with the previous one. We do the questions at the end of our session, but I allow myself one overarching question maybe. Could you put your system in the complete arsenal of drugs, which they actually are not so many, are used for the treatment of osteoarthritis? If you have a 2 months bioactivity with your systems, how does this fit with the clinical situation? Of course, it's also in one joint, if you have more joints which are infected and so on. Can you comment on this?
Yeah, so I agree. For inflammation, if you have several joints, then it may be in a clinical setup more complicated to inject several joints. Here we have a local approach, definitely. So we need to have a disease which is mainly at one joint. This is the case for osteoarthritis very often. So we had this in mind, and that's why I didn't talk about arthritis itself, but more about osteoarthritis to treat one joint in an excellent manner. And the intra-articular injection is a very attractive way of doing, albeit with the problem of not being repeated so often. So I think that given this context, for drugs which are amenable to this kind of technology, which has a poor solubility, for instance, this extended release might be beneficial for this specific patient which has disease at a specific joint.
Thanks so much. And we proceed to the next speaker. We all know her, Viola Vogel from the ETH, and the topic is, of course, now again, Mechanobiology. Please start your lecture, Viola.
Can you see my screen?
I do.
Thank you very much. So the lymph node is a real wonderful meeting place of immune cells, T cells, B cells, dendritic cells, but then also of fibroblast-like cells that constantly remodel and maintain the infrastructure of a lymph node. And as we try to understand how to combat infections more efficiently, inflammatory processes more efficiently, we are also looking at, in the moment, lymph node and lymph node tissue, and whether there is some knowledge that we are lacking right now in terms of how lymph nodes function. And there are quite interesting discussions certainly ongoing in the context of COVID-19, whether or not, and what is the role of T cells in COVID-19 infection, why immunity is about more than antibodies, something and an important question that we need to ask right now. And here, if you like to search for that, is an article just published from a group in Oxford. And what is important to notice is that lymphopenia, and that is low levels of lymphocytes, is a main feature of COVID-19 infection, affecting CD4 and CD8 plus T cells, B cells, and it is much more pronounced in severely ill patients. T-cell responses in severely ill patients may be impaired, overactivated, or inappropriate, and further research is required to elucidate this and inform in order to improve on treatment strategies. So a similar theme to the theme that Nikhil discussed in the context of macrophages. So here's a sketch that you will find in the good or typical reviews about lymph nodes, where you have areas in the lymph nodes where B cells mature and get trained. You find zones rich in T cells, but in between is a very dense network of extracellular matrix. And most of the work in the past focused on the role of collagen and collagen conduits in the context of priming, but then also staging inflammatory responses. And there is a cytokine that is the only cytokine that is known today that does not activate, but that tames immune cells. But most importantly, it is important, its presence is important in order to mature immune cells. And so it was asked already 10 years ago whether one can harness this particular cytokine for immune therapeutic applications. What we know today is that IL-7 promotes B cell and T cell proliferation and maturation. And the lack of IL-7 causes immature immune cell arrest. And IL-7 is much more potent in an extracellular matrix-bound state. And that is something that is most commonly neglected right now in discussions, but also in the literature. And I would like to tell you a little why we think this might be a really important piece to the entire puzzle. So based on what I just mentioned, Interleukin 7, IL-7 is also explored for therapeutic applications to treat severely ill COVID-19 patients. And what you see here is a treatment of IL-7, and what you see is the lymphocyte count is function of time after the treatment begins. And you see that the lymphocyte counts goes significantly up in severely ill patients after 15 days of treatment. So the request is also there in the moment to allow to use IL-7 in the context of the clinics right now. So question, how does it work? And are there any insights that might help us to improve IL-7 based treatments? So whilst IL-7 binding to the extracellular matrix may be really important, IL-7 concentrations in blood, but also in the lymph are incredibly low. They are in the picogram per milliliter range. They are produced by lymph node fibroblastic reticular cells. IL-7 regulates and maintains lymphoid functions. And B cells and T cells, dendritic cells, lymphatic endothelial cells, they all compete for a molecule that exists in such low concentrations. So what we know today, though, is that IL-7 is much more potent in the matrix-bound state. So we started looking into its interaction with fibronectin. What was known was that it binds to fibronectin, but the binding site was not known. And in a series of studies that we have published recently in Nanoletters, we were able to identify crucial binding site, which is located on the type 1, the second type 1 domain of fibronectin close to its N-terminus. Here you see the structure of IL-7. This upper part binds to the receptor, but then you see a long loop, and this long loop binds to the type 1 domain. So while lymph nodes are not just rich in collagen, you see on the left side a stain showing that healthy lymph nodes are very, very rich in fibronectin as well. And a question that we were asking was, is IL-7 binding to lymph node extracellular matrix fibers dependent on the degree of stretching of these fibers? Because we thought the stretching might change if you swell a lymph node. So on the right side, you see simply how cells are able to stretch matrix fibers. What you see are fibroblasts. They're sitting in a cell-derived extracellular matrix. You see fibronectin in green, and you see how cells that are in such a network can constantly stretch and let go, stretch and let go these fibronectin fibers. But how can we measure the fiber strain in lymph node tissue? Are there methods that we can exploit to do so? Because so far there were no sensors that allow us to measure fiber strain, not rigidities, but fiber strains. And we made a discovery a couple of years ago. And by looking into how bacteria bind to fibronectin and fibronectin fibers. And many bacteria, including S. aureus, they have long peptides on their surface. And these peptides were known to bind to fibronectin. Again, they bind to the type 1 domains. The type 1, 2 was the one where IL-7 binds. But this peptide binds to several in a row. So it's a multivalent binding motif. And when we simulated that interaction, we saw that if you stretch the fibers, the bacterial peptide pops off. So with that, we recognized that we can maybe utilize a peptide as a nanoprobe to measure the tensional state of fibers. We engineered a peptide that binds with nanomolar affinity to fibronectin fibers, but only if they are relaxed, not if they are stretched. And therefore we can stain tissue in a similar way as what we do in immunostaining. So in order to validate that this peptide really works well in cell culture, we use 2 probes. In the past, and I reported about that before at the beginning of CLEANUP, we developed a FRET probe where we label fibronectin with donors and acceptors, and as fibronectin gets stretched, the FRET ratio is decreasing. The efficiency of energy transfer is decreasing. So we have 2 probes now that should both be sensitive to the tensional state of fibronectin, the one measuring the entire range of changes. And so we plotted the FRET ratio of extracellular matrix fibers on one axis, and then labeled the peptide with a 3rd color, and saw indeed a very nice correlation, which you also see in the images. Below you see extracellular matrix produced by fibroblasts, and you see the FRET ratios. The red areas are structurally relaxed fibers, and the greenish blue are highly stretched fibers. And what you see is that the peptide indeed binds to the relaxed areas. So with that, we now have a peptide that can detect the fiber strain. And we therefore, and since no information at all is available, how much fibronectin fibers are stretched in healthy organs, we first assessed fibronectin fiber strain in healthy organs. We, for that reason, utilized, first of all, a polyclonal fibronectin antibody, and saw that all the healthy organs that we studied are rich in fibronectin, which is known. But then we, again, photo-labeled our peptide probe with the red color and saw very few regions that light up in red, indicating that fibronectin fibers are maintained in a rather high tensional state in healthy tissue. When we then asked this question about lymph nodes, we were very surprised to find that fibronectin fibers are already highly tensed in the naive lymph node. You see it mostly green with very, very few peptide areas or areas that bind the peptide. Then if we just swell the lymph node with adjuvant, what was surprising for us was the fibronectin fiber tension was still high. So that there's no major difference between the fibronectin fiber tension between a naive lymph node and a highly swollen lymph node. But then if we virally infected the mouse and then sliced the lymph nodes, what you see is major areas where the lymph node is defected, major areas where the peptide is binding. If we then do high resolution imaging of the lymph nodes, we confirm that they are that the peptide mostly binds only in virally infected lymph nodes, indicating what has been shown already in the sketch on the left side is that the virus infection tears apart the fibronectin fiber network, and therefore fibronectin is structurally relaxed and binds the peptide. But why is this important? Why should we even care about the tensional state of lymph nodes? And in order to ask this, we asked whether IL-7 binding to fibronectin fibers is regulated by the tension of the fibers. In order to do that, we use these type of pulling assays where we pull fibronectin fibers out of a droplet. We deposit them on stretchable substrates. And we could indeed previously shown that stretching destroys some binding sites and others get activated. So in what I show you in the next few slides, we can show that IL-7 binding to fibronectin fibers is upregulated when we stretch the fibers. How did we do that? We deposited, as you've just seen, these fibers on stretchable substrates. We then added small amounts, trace amounts of our thread probe to the samples. And indeed, as we stretch the fibers, you see an upregulation of IL-7 binding to these fibers. Why is this significant? IL-7 binds more to stretched than to relaxed or enzymatically cleaved fibronectin. Why is this significant? We looked into the literature because it implies that perhaps if you tear lymph node ECM fibers apart, that IL-7 is released from the lymph node. And in order to see whether there are any indications that this might be the case, we looked into the literature. And I indeed found a rich literature on HIV infections and all reporting that upon HIV infections in human, the plasma level of interleukin 7 are increasing. But it was often not attributed to release of IL-7 from extracellular matrix fibers, but people thought IL-7 production in the body is simply upregulated to increase the levels to that degree. But people also found if you do an IL-7 therapy, you can increase the absolute number of circulating CD4 plus T cells. So obviously IL-7 is lost in the lymph node and you have to supplement it in order to keep the maturation of immune cells intact. So is the destruction of the fibronectin ECM fiber network in lymph nodes part of the big puzzle? And can it, in part maybe, explain what has been observed in the literature? So if we look at, and come back to the beginning of the story, at COVID-19, IL-7 is considered to be used as therapy. I've shown you some data, how the counts on lymphocytes goes up with IL-7 treatment, but you also notice that the IL-7 concentrations that are administered are excessively high compared to the native concentrations that you would find in blood plasma or in lymph nodes. And we hope that knowledge about the role of the mechanobiology of extracellular matrix might in the future make a contribution in better understanding lymph node functions, but again, also how we can maybe utilize that information to better intervene. With that, I would like to thank you for your attention. I would like to thank all my current and previous group members for their wonderful work. I acknowledged the members with pictures as we went through the story. And I would also like to thank the funding agencies for generous support over the years that made this work possible. Thank you very much.
Thank you, Viola, for also a very eye-catching, actually, for me, results. Very innovative, I would say. And yeah, we can discuss about the implications of your work at the end of this session, over 20 minutes or so. For now, I want to have a 1-minute discussion with you. I was also intrigued by the difference between adjuvant-mediated swelling of a lymph node and a viral infected lymph node in terms of the effects you see regarding stretching. And so the adjuvant is only giving more fluid and that's it? Or is the adjuvant, can you explain that difference?
So adjuvant causes, results in a major influx of fluid, but also invasion of cells. The reticular fibroblasts are sitting on really rigid collagen conduits, and it's on the surface of these conduits that they assemble fibronectin matrix. And this fibronectin matrix is stretched already. We know from cell studies on solid surfaces that they stretch fibronectin fibers highly if they are sitting on rigid surfaces. So these conduit surfaces seem to cause that the reticular cells assemble fibronectin fibers in a stretched state already in the naive lymph node. That's our explanation right now. Okay, yeah, it's an acceptable explanation for now. Of course, it's always difficult to exactly define what's happening, but really interesting. For now, I would like to continue over 20 minutes or so. For now, we proceed to the last lecture, and that's a lecture of, and thank you, Viola, for doing this. And that's a lecture by Professor Carlos Mota. He's from Maastricht, so the Netherlands. He's close by. And he will talk about a nanotechnology platform for tympanic membrane regeneration. Please, Professor Mota, take the floor.
Thank you very much. I would like to also thank Gert and all the organizing team to put this conference together on unusual times. So today I'm going to speak a bit about an European project that we have looking at strategies to regenerate tympanic membrane perforations. So this is a bit the outlook for today. I'll discuss a bit tympanic membrane and what are our strategies to come up with alternatives to regenerate the membrane. So what is the tympanic membrane? The tympanic membrane is a very small membrane that separates the outer ear from the middle ear. It is responsible to capture sound waves and converting them into mechanical motion that is then transmitted to the cochlea. to be converted then into electrical signals so that you would have the hearing capabilities. Normally, the tympanic membrane can be affected by chronic inflammation or trauma. And when, for instance, chronic inflammation occurs, in the cases of cholesteatoma, the membrane can rupture and then you have a perforation that can be persistent. So in these cases, normally what the clinicians use is autologous tissue, normally temporalis fascia or cartilage. So normally this autologous tissue is placed either on top or underneath the tympanic membrane. And as you can see here in this example, this is the picture of using cartilage. But what normally happens is that when you use cartilage or even temporalis fascia, there is a mechanical mismatch of the tissue, and there's also a limited regeneration of the tympanic membrane. So you'll never hear with the same level of capacity as in the healthy native tympanic membrane. So as you can see here, in the case that cartilage was used, there's also a limited remodeling of this tissue that is implanted on site. So if we look at the tympanic membrane more in depth, we have a microscopic size of the tympanic membrane is around 10 millimeters, and it's a bit concavous. Also to have better efficiency of that sound conversion of the sound waves to mechanical motion. So if you look a bit in detail, the tympanic membrane is divided in 3 layers: cellular layer and epidermal epithelium, lamina propria, which is highly composed of collagen fibrils arranged in a very peculiar form, either concentrically or radially, and then we have the mucosal layer as well. So all this cross-section is roughly 100 microns, so this is a very packed collagen fibril network. So back in 2015, we started to look on biofabrication techniques to produce temporary scaffolds to fill in these perforations on the tympanic membrane. So on the most severe cases, we have to replace the full tympanic membrane. So what we used was initially 3D printing and electrospinning, two of the biofabrication techniques that we normally use in the labs, to create this temporary scaffolds. So scaffolds of biocompatible and biodegradable material. With the 3D printing techniques, we wanted to give that functionality or mimic the functionality of the collagen fibrils. And then the electrospun mesh is normally something that is very useful for you to seed cells and also to have that three-dimensional network replacing a bit the tympanic membrane effect. So the limitation at this stage was that there was some technology limitations. For instance, to give you an idea, the filaments that we could produce at that time of 3D printing were around 300 microns. So this was normally an implant that was rather stiff. So we wanted to optimize that. So we were a bit lucky on getting some funding to study a bit more in depth. So what we wanted to, and what we are doing still on this project, is to look at 4 nano dimensions. So we are looking at incorporating some nanocomposites on these membranes, and namely nanochitin. For the sake of time, I will not show a lot of results on that today. We want also to incorporate nanoparticles for controlled drug release, also because in clinics what is used is normally droplets of the drug or collagen sponges soaked with the drug, and that's not really efficient. So we wanted to incorporate nanoparticles in our implants. We wanted to optimize the fabrication, also in view to optimize then the nanovibration response of these implants upon implantation. So we did a lot of optimization, looking also more specifically on the geometry of the tympanic membrane and trying to mimic it. So we did some computational modeling, we optimized the manufacturing techniques, and then we did a more thorough characterization of the mechanical, acoustic, and biological properties of these implants. So this was a Python script that we had to develop to create a bit the 3D printed patterns of our tympanic membrane implants. So as we can see, we selected 3 distinctive cases for our studies: case 1 where we have just concentrical fibers, a case 2 where we have radial, and a case 3 where we have radial and concentrics. Of course, this is not a full replica of the collagen fibrils because of dimension, but what we want to do is also to make a bit of a stability effect of the electrospan meshes that we use underneath, so that the surgeon can also handle them upon implantation in the future. So we did some computational modeling also to try to understand before manufacturing the different cases, the effect of the different microstructures. So on the case 3, of course, we would have a bit more reinforcement, so we would have a young modulus higher than the other cases. And we also simulated the acoustic vibration of these membranes according to the different patterns. What we can see here is that according to the different reinforcements of the microfilaments, then we have a first resonance frequency, which is one of the benchmarks to understand the quality of the implant. We look at the first resonance frequency, and as we reinforce the structure, we have a higher resonance frequency for the implants. So on the fabrication, we try to optimize a bit the fabrication strategy. We try to go on the nano or submicron, if you want, size of the nanofibers. So there's a lot of processing parameters that we optimize, for instance, the polymer concentration, the polymer that we can use, the electrical field that allows us to generate these submicron fibers. So by playing a bit with the parameters, we can generate fibers of around 500 nanometers. And then the extrusion patterns with the 3D printing technique that we use on top of these electrospan meshes, we also wanted to optimize it because in the total implant, we wanted to have a thickness of around 70 to 100 microns. So by optimizing again a bit the 3D printing technique, we can produce fibers of around 50 microns, and then with the combination of the electrospan mesh, we are normally below this 100 microns of threshold that we were aiming at. We did some mechanical characterization of the meshes. This is not a really representative of the native environment, because on the back of the tympanic membrane, you have the oscillar chain, but of course that's very difficult to mimic. But here what we wanted to see was again the young modulus of this pattern. So we see an increase of the young modulus, pretty much what we simulated in the beginning, but we are still a bit below the native tympanic membrane, the native human tympanic membrane in terms of young modulus. But we believe that we can achieve the same young modulus as soon as we have the cellular component in there to contribute to the properties, mechanical properties of the membrane. We did also acoustic characterization of the different cases, especially looking at the first resonance frequency and the different extent of the influence of this three-dimensional printed patterns on top of the electrospan mesh. And what we can see is that the first resonance frequency on the case 3, which is the more reinforced, normally is a bit higher than the first resonance frequency of the native human tympanic membrane. There are a lot of reported numbers for the human tympanic membrane, but this is also normally tested on cadaveric samples, so it's not really very accurate, but we are more or less in the same range of the human tympanic membrane. Did some preliminary biological characterization. We are looking at creating this implant as an advanced therapy, but also as a medical device, so with or without cells. The strategy here is that we normally seed cells. In this case, there's also not a representative cell apart from primary cells from the human tympanic membrane that we can use. So that's also the difficulty of having an advanced therapy. But we use mesenchymal stromal cells that then we try to differentiate more towards fibroblastic type and also dermofibroblasts. And we see a very nice colonization of the full implants after 7 days. Yeah, on the initial time points, then we have a bit of a less uniform dispersion of the cells, which will then impact the outcome in terms of collagen deposition, for instance. So we also wanted to see how the cells would behave on the combined microfilaments and the electrospun mesh. So we see that when we have the microfilaments, then there is a bit of a densification of the cells around these microfilaments, kind of mimicking a bit the sieving effect. And these will be also the areas where we have an overexpression of collagen deposition. So as you can see here on the collagen, and I have on a couple of slides a bit of a more comparative case, we have more collagen being deposited around the microfilaments, which in our view is optimal for mimicking a bit also the collagen that is found on the native tympanic membrane. Of course, that we cannot densify a lot of these patterns so that we have more collagen. So this is something that we are trying to look into other manufacturing techniques that will allow us to do that. But our idea is to have the cells then being aligned on specific patterns that we have, then ultimately the collagen fibrils being deposited in the way that we want. So as you can see here, this is a specific collagen staining. On the controls, we have a bit of a random orientation of the collagen fibrils. But then gradually, as soon as we play with the patterns, then we can play a bit with the collagen deposition. It's a bit cellular dependent. So on stromal cells, on mesenchymal stromal cells, we have a bit of more impact of the microstructure on the collagen deposition rather than on dermal fibroblasts. So we are also looking a bit more in detail and also to try to quantify spatially what is the influence of these microstructures on the collagen deposition. So as I told in the beginning, we want also to include nanoparticles for controlled drug release on these membranes. What is used now in the clinics post-surgery is that the surgeon fills in the ear canal with a collagen sponge normally, or a gelatin sponge that is normally soaked on an antibiotic solution. And this gradually is released on top of the implant. Or if the implant is already starting to be regenerated, normally the sponge gets removed over time, and then the patient has to use eardrops with the antibiotics. So we want to use antibiotics loaded on our implant. So we started with we have a company in the consortium that has two proprietary reactors to produce nanoparticles with nanoprecipitation or double emulsion. And we have, these are controlled particles with gold or iron for us also to try to localize where we are locating these particles on top of our implant. And then we also loaded some drugs on these nanoparticles. So the particle size, we use two polymers, PLJ that is normally frequently used, and also PCL. And we produce particles and particle sizes around 200 nanometers. We have different loading efficiency on PLJ and PCL. On PLJ, we can load up to 60% of our initial drug concentration, and on PCL, we can load up to 80%. The difficulty here is that we have very fast release on these nanoparticles, so we are also looking into ways to overcome this. So normally on microparticles, we can then extend the release profile, but we are mainly interested to have a sustained release for the 2 initial weeks. But as you can see here in this graph, the nanoparticles, the drug release on the nanoparticles happens almost in 24 hours in its completion. But they are, we have tested them on the gram-negative and gram-positive, and they have comparable inhibition with the free drug as well. So we try to think of an easy strategy to load these nanoparticles on our electrospun meshes. An easy way would be to immerse the electrospun mesh or to dip the nanoparticle suspension on top of our electrospun mesh. On the double emulsion, we have a lot of surfactant films being deposited on the mesh. So this is something that we also don't want because we also want to have the cells being able to colonize the full cross section of the electrospun mesh. And then on the nanoprecipitation, if we do an immersion approach, then we also have a lot of clusters of nanoparticles, not really a uniform dispersion of the nanoparticles throughout the mesh. So what we did was to try to optimize this dispersion of the nanoparticles. So what we did was a combination of electrospinning, where we create these submicron fibers, and then at the same time electrospray the nanoparticle solution throughout the construction of these meshes that have more or less between 50 microns to 100 microns in thickness. So on the double emulsion, again, we have the surfactant influencing a lot our electrospinning process. So we also have a bit of a thickening of our submicron fibers produced by electrospinning. But then when we use the nanoprecipitation, we have a better distribution of the nanoparticles, and we have also a better control on the submicron filaments that we can produce with the electrospinning mesh. So we tested the stability of these nanoparticles on the mesh. So after day 3, in this case, we still see the nanoparticles attached to the mesh. We believe that this attachment is promoted by the effect that when we create these submicron fibers with the electrospinning, there's still a bit of residual solvent, so the particles will be able to adhere to the electrospun mesh. So this is good. This is what we want, because we don't want to have the nanoparticles being released without releasing first the drug. We also tested the cytotoxicity. The nanoparticles are not toxic, and we can see that the cells colonize the full membranes with the nanoparticles. What we are currently doing is that we are trying to optimize also the load of these nanoparticles on the electrospin mesh, because we need to know exactly the amount of nanoparticles so that we have the amount of drug that we need for the 2 weeks to be released in a controlled way, but also with the MIG value that we would need to inhibit bacteria proliferation. So this is in a nutshell what we have achieved. So we used advanced bifurcation techniques to produce electrospun meshes with a proper thickness that we aim for, mimicking a bit the thickness of the human tympanic membrane. We also have optimized the electrospinning technique to go more towards the nano-sized fiber mesh. It's challenging to go to really nano-size with the electrospinning, because there is also a proper balance. So what happens normally, you can produce nano-sized fibers, but normally you have a bead effect happening. We optimize also the mechanical acoustic properties of these implants. We did some thorough characterization on the acoustic parameters, also looking at if the values that we will be obtaining are near to the human tympanic membrane. On the biological characterization, we are looking more in depth on ways to control this collagen deposition. And then we also demonstrated that we can load the nanoparticles on these meshes in a controlled way, but we are looking to ways to have the amount of drug being released for the 2 weeks. So we are looking into hybrid solutions to optimize that. So I would like to acknowledge the full team, the consortium partners, and the funding. This is part of the Euro-Nanomed 3 of Eranet. And I would like to thank you all. Before I finish, I would like to draw your attention that we are organizing World Thermis here in Maastricht next year in November. It was initially in May, but we had to postpone it. So hopefully we will be able to see each other if you are participating in Thermis conferences. And I would like to thank you all for your attention.
Thank you, Dr. Mota. Also, I don't know if in the world people are working on tympanic membrane implants, or that you are one of the few groups, or maybe only group in the world. Can you reply to my comment?
Yeah, so there is several groups. There is more groups looking at soluble growth factors to regenerate small perforations. So if they are detected early on, for a big full replacement of the tympanic membrane when we have a severe perforation, there's a couple of groups for 4 or 5 groups looking at biofabrication techniques to regenerate the tympanic membrane. There is a group in Harvard. We were actually one of the first ones publishing these approaches on biofabrication. But yeah, this has drawn a lot of attention. So there's more groups nowadays looking into it.
Well, it would be very great if this disease, like otitis media, I think it is, could be served and people suffering from this. We are now at the end. Thank you for this talk. All the 4 talks will be discussed now. That was the idea. And there are no questions yet, I see, to the last talk, but I will refresh soon and then probably also see questions for the last talk. But let's start with the first lecture of Dr. Jain. And may I ask you, Dr. Jain, there is a question from Shiki Wang from the University of Helsinki. Is it possible to convert the macrophage to M2 phenotype by confinement of mechanobiology methods?
Okay, yeah, that's a fantastic question. So yes, it is. And it was published by Wendy Liu at the University of Davis. And if you remember, what I have shown you is that when macrophages enter the process of M2, they become this really polarized cell and throw these finger-like projections. So what they have done is they generated these microstripes of really long lengths and they culture macrophages on them. And macrophages actually sense those stripes, become really polarized, and surprisingly, even in the absence of any kind of chemical signals, just by giving them these right geometrical or morphological signals, they were able to convert M0 macrophages to M2 macrophages. So it's pretty much possible, just by mechanical forces.
Well, that's a very clear and optimistic answer, that this might be something. Then there is another question for you.
Yeah.
This question, the questioner is not known, but the question is, as both M1 and M2 polarized states show a change in morphology, could actin inhibition also affect M2 polarization? So actin inhibition.
Okay, so our understanding is that M1 inhibition, the actin inhibition definitely inhibit M1 activation, but whether the same happens for M2, we don't know yet. Because, and it's actually very nice suggestion also that we should check what is the status of actin, the organization and the levels of F-actin in M2 condition. I am not sure whether anybody has tried it, but I'm sure that it's not very difficult to do it. So the short answer is right now, we don't know.
Okay, very clear. A third question for you is from Priscilla Shilrev. And she asks, is it possible to compare the results of the macrophages confinement experiment with the confinements cells have in tissues or 3D organotypic models?
Okay, now that's an expected question from, I'm pretty sure the person is from immunology background. Indeed, as Viola has shown in her presentation, that in tissues, cells do feel these mechanical forces. And as we have also shown only in maybe in 2 dimension and quasi 3 dimension, that cells do feel these forces and respond to them. And I'll just give you one example. If you take a tissue section from Alzheimer patients, or if you look at the neuroinflammation, you can precisely and very clearly show or see over there that the macrophages are really confined, and that confinement has a direct role in their activation. A recently published paper in Nature Medicine has also shown the same effect in granulomas. So in solid granulomas, you have a cavity surrounded by macrophages, and macrophages are confined over there. And that confinement has a direct influence on their activation. That's one of the few of the examples where we can see that the similar kind of behavior we can see in vivo, but yeah, we definitely need to do more.
Okay, well, thank you. These were the questions for your lecture. I don't know if some other questions will be added. I will refresh a little later. Maybe one of the lecturers would like to ask a question to you. No, then we go to the lecture of Olivier Jordan. I could not see any added question by the audience in my list. Maybe it still comes, but I refresh later. But I just wonder, you are putting nanocrystals in the polymeric particles, I should say. Did you compare, so how is this sustained release process? You have an effect over 2 months and the drug is slowly released over time. But did you also test other forms of the drug inside the particles? So not a nanocrystal form, maybe you have encapsulated in an, maybe you could even put a liposome in there or whatever, an other form of, so that you can also, that you can maybe state that nanocrystals are really superior than other forms of the drug.
Well, it's a question of feasibility. I mean, encapsulating a liposome into a macroparticle of polymer, well, it's a nice idea, but it would be a bit more complex in terms of process. The main idea is to, you know, with nanocrystals, you have a fast dissolution, you have a lot of available medication on the market like this, which have an effect over days maximum. At least in terms of dissolution, of course, this therapeutic effect can last longer. Now, the nanocrystal sees its environment as a sink condition, so the dissolution is very fast. The driving force is a gradient of concentration. If you put a nanocrystal into a polymer particle, then the gradient is a difference with the solubility of the drug in the polymer. So by this way, you actually slow down the dissolution of your nanocrystal. So it's just a way to change your environment as nanocrystal in order to modulate and to slow down its release. And that's how we can have release over several months with this nanocrystal. Nanocrystal is a very opportunistic manner of putting very high drug dose because it's a very high packing that is difficult to achieve with conventional encapsulation techniques. As for comparison, we made some comparisons, for instance, with, I did not show you the result with an anti-inflammatory MAP kinase inhibitor, pH38, where we compared actually in over 1 or 2 weeks in an inflammatory model this time, liposomes with these particles, and clearly with liposome, we had a strong effect, but on a much shorter duration than with these particles. So I think it's a really unique benefit with those particles, the downside being the limitation of the local administration, of course.
Yeah, of course, but in some cases it can be also really helpful to do. And I think there's really a clinical need for such formulation you have. Did you also correlate drug levels with bioactivity? So the 2 months bioactivity you see, did you also measure drug levels?
No, we did not. We could not, unfortunately, in mice do PK in the articulation in the joint in the plasma due to relatively low availability of blood. We made a lot of different analyses, including ELISA, showing VEGF, downregulation, and so on. That's something we need to do. I mean, the PK itself on the local side and the animal side, but probably with animal model, which would be of a larger size.
Okay, thank you. There are no questions for you added in the question list. So maybe the speakers would like to have a question to be addressed by you. No. Then we go to Viola Vogel. You have a question in, let's see, go to the question list. There are actually only 4 questions in total for this session. And this is from Jay Prakash of the University of Twente. It's for you, Viola. It's tracing fibronectin in tumor stroma. He's interested in that and for his research. So have you used the peptide probe to examine the status of fibronectin in tumor?
This was actually the first experiment we did before turning to the lymph node. And also in tumors, since people have studied the Young's modulus of tumor tissue compared to normal healthy tissue, seeing a major change. But people also showed that cross-linking is taking place. So why are certain tumors more rigid And is that connected with increased fiber tension of the ECM? So the very first tissue that we stained was tumor tissue from mice. And we saw that in those cryo sections that a lot of peptide is binding, but very heterogeneously. In some areas, nothing at all. We started studying whether there are correlations between whether these locations are close to blood vessels. There's no correlation. We see major correlations with regions that show second harmonic positive collagen fibers, really thick collagen fiber bundles. So we are wondering whether either the tension that cells apply is applied then to the collagen bundles and that fibronectin is therefore relaxing, or what we also find is that these regions where we find relaxed fibronectin fibers are very rich in myofibroblasts. And we always thought myofibroblasts would enhance the tension because they have upregulated contractility, but they also produce a lot of ECM components and certainly other also enzymes. And we think that either it's the high collagen content in these areas that causes fibronectin fibers to relax, or enzymatic cleavage that happens there. So we did that now with a number of different tumor mouse models and are studying human cancer tissue right now.
Okay, that's also hopefully clear to Dr. Jay Prakash and all of us. Yeah, but I would like to talk with you a little bit about the implications of your work in more general, because matrix, fibrous tissue, and actually occurring in many diseases, fibrosis. Myself, I'm more going direction also to NASH disease and liver fibrosis. And could your systems, how do you foresee the future if you make a step to COVID? It's very timely, of course, but if you make a step to these also, for instance, NASH disease, there are hardly any therapies. So how do you see this?
So a question that we are asking right now, and therefore the benchmarking of healthy organs was so absolutely crucial, in particular that the fibronectin fibers under high tension. We look into a number of different diseases right now, fibrotic diseases included. And okay. So we have some indications in the moment that the peptide binds to regions where extracellular matrix is diseased for all kind of different reasons, whether it's caused by inflammation, whether it's caused by fibrotic transformations, whether it's caused in tumors. But we need many more data to substantiate that this is indeed the case. And certainly we would like, and we have set up now a number of clinical collaborations in order to get away from mouse models only, because we know in mice, the tumors are induced, they grow very rapidly. The extracellular matrix probably has very different qualities to the qualities that you find in an older person, where these diseases start gradually and are advancing gradually. And so we want to also ensure that what we see in young induced mice diseases applies to humans. Mm-hmm. And since nothing is known about the tensional state, we need to make sure that what we are observing is significant and reproducible and not dependent on a single mouse model.
Thank you. Yeah, you clearly indicate it's still a long way to go, but it's fascinating, I must say. Thank you. There are no other questions. Maybe the other lecturers would like to ask, they can just unmute. But if not, then I would like to end up with the lecture of Professor Mota, because there was the word nanoparticles in there, so I cannot let them go without any question. There are no questions from the listeners to you, but my question is actually, I missed, what was the antibiotic you put in this?
Yeah, so that's also a bit of a tricky question for us to, we included ciprofloxacin, but in Europe, pretty much every autologist will have his preferred antibiotic. But the ciprofloxacin is commonly used antibiotic in most countries. So that's our choice.
So that makes a sort of logic choice to at least to start with this. Yeah. I agree. Could you maybe benefit of the technology of Olivier, Jordan? So
so we had a bit of more success on microparticles actually. So we had a longer release and a bit of a less burst release. So that's something that we looked into. The difficulty there is then that we will have much lower capacity of loading, and that will also change topographically our implants. So that's something that we would definitely look into. But yeah, maybe I can drop an email to Professor Olivier to see if we can brainstorm a bit around that.
So that would be a fruitful benefit from this session. I have also another question. You say if you have these nanoparticles, you put them in the air then on the implant. So what is the adherence mechanism? Or I can imagine that the mobility of nanoparticles will also not be very high in that environment. But how do they adhere to the implant?
Yeah, so here, since we were combining the two techniques, the electrospinning and the electrospray, we believe that when the electrospinning fibers are drying out, because we start from a polymeric solution, that there is a bit of a contact fusion between the nanoparticles and the electrospinning mesh, because there's still a bit of residual solvent from the electrospin mesh. So that's why we see them that they also stay there for longer for the culture period. A bit of our concern is that there should be a threshold of residual solvent that then will burst the nanoparticles. So that's something that we are also trying to look into, especially with TEM, and that's why we also synthesize the particles with gold or iron in the core, so that we see if there is at that particular contact point, if there is a bit of the disruption of the nanoparticles. But that's something that we are still looking into.
And thank you. And then a last question from my side. If others of the speakers have a question, they can after me address it. But I was a little bit, but maybe I do not understand it. It's too difficult for me. Many things are easily very difficult for me. But it's about the young modulus of your implant. You say it's not that, it should be improved by adding this cellular component. How is the cellular component then improving this young modulus? Because it's not the same as in humans.
Yeah, so what we see is that there is collagen deposition. So then the collagen will reinforce a bit the structure. But of course that we don't have the implantation case, right? The implantation case would be that your collagen fibrillar deposition will happen also with the surrounding ear canal. So that's where the young modulus then will probably increase as soon as the regeneration starts. So what we want is that the implant has the minimum properties to facilitate also this growth of new tissue on the top and on the bottom. So then as soon as the implant degrades over time, then the new tissue will be the predominant contribute for the mechanical and acoustical properties. So that's why we are trying to aim for more or less the mechanical properties that we see in the tympanic membrane. But of course, that the ideal scenario is difficult. As soon as we have regeneration, then we expect that then we will have a bit of an improvement on the mechanical and acoustical properties.
Okay, very clear. Thank you. Well, are there
I ask a short
question? Yes, of course.
To what extent do you observe scarring?
That's something that we would like to see, but when we start the in vivos. So the difficulty of the tympanic membrane is that the in vivo model that is available is only a chinchilla model. and there's only a couple of groups in the world that do the chinchilla model. So that's something that we will probably look into. So as I said, we want to use this as an implant as a cell-free approach, but also cell-based approach. So the idea is that on the cell-free approach, then the cells from the surrounding tissue will migrate towards the implant. So that's ideally we would not have a lot of fibrosis in there and scarring. But that needs to be seen on in vivo. On in vitro, I think we will have difficulties on seeing that.
Because very often the cells can't really migrate into the materials because there are too many fibers and then only migrate on the surface. So how do you design the scaffold in a way that the cells can really effectively infiltrate and rebuild the matrix?
Yeah, so on the porosity, so the electrospun mesh component is highly porous. The pore size is normally much smaller than the cell size, but normally what we see is that the cells can stretch and push the fibers and go inside and across the membrane, because there's also a very thin membrane. So normally the electrospinning meshes are known to have a bit of a barrier effect as well, so that you have a monolayer, but then the cells will not infiltrate. But if you create it thin enough, and our membranes are below 70 microns, you still have a very good diffusion of nutrients, so you'll see that the cells will also proliferate inside and then gradually degrade the implant.
Very good.
Thank you. I think
there's a question from
one question more, but it should be a short one because we should really finish by 12:30. What time is it? 4 minutes.
Just getting back to the antibiotic story. So did you consider actually using the fiber itself for the delivery, that is loading the antibiotic into the fiber?
Yeah, so that's what we are actually looking at now, that we would have a combination of them.
Do you hear me?
Yes. Yes, Professor Beat.
We will end in a minute. Yes.
Should I continue? Okay. Yeah, we are looking.
Do you hear me?
Yeah.
Yes.
Yeah, we all hear you.
Gert?
Yes, we all hear you.
We need to
finish
the whole for Anil Patri now.
Okay. We will end the session. So maybe one sentence as an answer.
Yeah, we are looking into loading the antibiotics on the electrospun mesh as well, but the degradation rate might impair. So we might have more than 2 weeks release profile there, so that we are looking into that.
Okay. Thank you. We have to stop. Thanks to all the speakers. Thanks to all the listeners. I enjoyed the session, and you see we always have never enough time for great science. We stop now. Hope to meet you virtually or in live later in life. Thank you so much.
Thank you very much.
Bye-bye.
Bye-bye.
Bye-bye.